Did you know that the brain contains chemicals that can affect the brain function and affect our thoughts? These chemicals are known as metabolites and can be non-invasively detected using MR spectroscopy, which is a type of MRI sequence that can help display and quantify the metabolites in our brain. In this way, we can interpret the patterns of the chemicals in healthy brain compared to disorders such as Alzheimer's disease, stroke, brain infection, and even brain tumors. Hi everyone, I'm Priya Supaya. I'm the founder of the Malaysian Society of Neurocognitive Clinical and Behavioural Imaging, known as NeuroCoB Society Malaysia. Today, in this webinar, NeuroCoB Society is collaborating with the Nuclear Imaging Unit of Hospital Sultan Abdul Aziz Shah, University Putra Malaysia, and METLiT.ai, a tech startup based in Seoul, Korea. Today, we have the honor of welcoming Dr. Hyeong Hun Lee, who is the Chief Technical Officer at METLiT.ai. He will present on the fundamentals of MR spectroscopy of the brain and introduce artificial intelligence techniques that can help to automate the quantification process of brain metabolites. I welcome Dr. Hyeong.
Okay, thank you for the introduction, Dr. Priya, and thank you for the opportunity to present about this talk. Once again, I'm Hyeong Hun Lee, serving as the CTO of the startup called METLiT in Korea. Today's talk will cover the principal introduction of MRS, data acquisition and processing, and lastly, MRS combined with artificial intelligence, which has been the hot topic in the radiology for the nearly decades, and those the scientific applications. So, comparing to MRI, MRS can be presented by this figure. So, comparing to MRI, MRS can be presented by this figure. While the MRI modalities provide anatomical and functional information non-invasively, as shown like this, MRS captures the resonance signal in a one-dimensional data format selectively from the specific volume of interest, for instance, hippocampus region. Through this, MRS provide both biochemical and functional information. Essentially, MRS data can be acquired on the same MR scanner that acquired with MRI data. So technically, again, this is what it looks like. First, MRI employs a pulse sequence that acquired signal from the specific slices, which can be done to fill the K-space region in the two-dimensional place. And subsequently, depending on the data dimension, MRI images are reconstructed using 2D or 3D FFT, enabling them to be utilized for specific objectives. On the other hand, MRS primarily uses the pulse sequence composed of 3 RF pulses and their paired gradients, targeting the signal acquired only from the particular voxel like this. This data is then transformed into the frequency domain using 1D FFT, followed by appropriate data processing for subsequent analysis. As such, MRS also has several sub-modalities. One is the single voxel spectroscopy, and more technically, there is spectroscopic imaging, which is a fusion method with MRI. And other specialized submodalities include fluorine or phosphorus MRS, and so on. Most of these submodalities within MRS should require further optimization of measurement steps or the establishment of new methodologies based on the specific research objective. And this process leading to reduced practical manner in medical field. Therefore, today, I'd like to focus on the single voxel proton MRS, which is the relatively straightforward and widely utilized in clinical side. So typically, When we acquire the proton MRS data from the brain region, we can observe approximately 20 different metabolites like this. These metabolites, unless associated with malignant neoplasm like brain tumor, usually present concentration ranging from the 1 to 20 millimole. When I write the observable here, it carries a different connotation from the quantifiable. This means that there are metabolites that can be observed but may be difficult or impossible to quantify. I will discuss this in more detail when explaining the MRS data analysis and later on. And each brain metabolite plays a different functional role within the tissue, brain tissue. For example, N-acetyl aspartate is frequently used as an indicator of neuronal activity. And glutamate and GABA serve as the excitatory and inhibitory neurotransmitters, and the glutathione plays a major antioxidative role in the human brain. Then, as we can see now, in proton MRS data from the brain, many metabolites resonate and appear within the very limited range. So, how can you distinguish each metabolite's resonance signal? We already have the answer to these questions. That is, the unique quantum mechanical properties, namely the chemical shift values and J coupling constant values, which originate from the molecular structure of each metabolite. Let's take a brief look at each part. First, the chemical shift is as you see now. The resonance frequency of the protons within each metabolite is entirely determined by each molecular structure. That is, the magnetic field strength that the resonating proton actually experiences is influenced by the electron cloud distribution within the molecular structure. Thus, even if the proton group are placed in same magnetic field strength, the resonance frequency is changed slightly due to the degree of these shielding effects. And it can be calculated as a relative difference compared to the reference chemicals like TMS, and this value is called the chemical shift. As an example of a brain metabolite in case of creatine, the proton in the methyl group, like this, is relatively isolated, showing the stable electron cloud distribution, and thus has a relatively strong shielding effect. Therefore, its resonance signal occurs in the outfield region. On the other hand, in case of the methylene group, it is located on the main backbone of the creatine molecule like this, causing the relatively scattered electron cloud distribution. And as a result, it produces a resonance signal in the downfield region. If the chemical shift represents a unique resonance frequency of proton attached to the single carbon or nitrogen, then the J coupling is another information representing a unique resonance signal pattern caused by the interaction between the nearby another protons. Skipping the quite boring quantum mechanical details, let's take lactate as an example directly. In lactate, resonance signal protons are located in the methyl group and the methane group. Unlike the creatine structure, these protons group are located directly by each carbon backbone, is leading to interference between the electron bonds and resulting in the splitting of resonance signal like this. The extent of this splitting is determined by the number of protons on the opposites of each group. Because of this, the signal from the methyl group is influenced by only one proton from the methane, resulting in the doublet pattern. Meanwhile, the methane group signal is influenced by the 3 protons of the methyl group, displaying the quartet pattern. At this time, the splitting intervals of all resonance signals show an entirely identical constant value, and this is called the J constant. To summarize, although the contents might have seemed a bit technical, the chemical shift and J coupling characteristics are critical properties for distinguish the unique resonance signal of the brain metabolites. And these characteristics for the observable brain metabolites have been well defined. So if you would like more details, please check the reference below. Next, let's see deeper into how MRS data can be acquired. As I explained in previously, MRS data is acquired using the same equipment as MRI scanner, MRI, known as the MR scanner. However, instead of using a single RF pulse like in MRI, MRS employs a combination of 3 RF pulses and their corresponding gradients. This forms what terms a pulse sequence, which result in the resonance signal from the specific area within the brain called the voxel. Unlike MRI, which achieve information on the 2D plane with the single slice selection, MRS pulse sequence involves a minimum of 3 slice selection to acquire the intersected signal. From this principle, variability of pulse sequence have been developed over time. The primary goal of developing this sequence is to minimize the signal distortion and amplify the MRS signal. The major MRS sequences can be seen in the diagram on the right and the table on the left, summarizing the several performance metrics of the pulse sequences primarily used in the clinical and preclinical research. Skipping the technical aspect and as of the current moment, the MRS pulse sequence most widely used in clinical and neuroscience field is PRESS, and thus further explanation will be based on the PRESS sequence. Now, we saw how the MRS pulse sequence operates. Let's assume that select a specific voxel location like this in the MRI image and conduct an MRS scan and obtain the data. And then the initial appearance of the data we get looks like this. And we can see here a very dominant single peaks appear in the MRS data. This because about the majority of human brain body is composed of water, as we already know. So the water signal also dominates in the MRS signal. If we expand the other chemical shift results based on the principles of chemical shift we discussed earlier, the signal of metabolites appears very tiny. If we expand the other chemical shift region based on the principles of the chemical shift we discussed earlier, the signal of the metabolites appears very tiny. This is because in the brain, the concentration of metabolites is very low level of few millimole, and therefore it is natural for the metabolite signal to appear very small in contrast of the water signal, which is distribute about 50 mole. So, what additional do we need to do to observe the metabolite signal clearly? In configuration of MRS pulse sequence, there's an additional component might be used called the pre-pulse module. As name implies, this module serves the purpose of helping us obtain MRS data more efficiently according to our specific objectives. Among them, a representative module is the water suppression module, as we can see now, which can be selectively suppress only the water signal resonance area. After the applied water suppression module, we can clearly see that the metabolite signals appear. And therefore, this water suppression module is fundamentally included as an MRS scan option in the MR scanner. Then, one question is the water unsuppressed data is unnecessary? No, it's not. Even though we primarily use the water suppressed data to quantify the metabolites, water unsuppressed data is essential for the pre-processing task that minimize distortion in the quantification process, as well as for the comparing the concentration of each quantified metabolite later on. Therefore, in conclusion, if we were to obtain MRS data from patients, we would need to both water suppressed and water unsuppressed data to achieve the complete results. Next, the important parameters for the MRS occasion are TE and TR, which have the same meaning as in the typical MRI sequence. That is, TR refers the time from the start of one sequence to the start of the next, and the T refers to the time from the start of sequence until the MRS signal starts to be measured. Both parameters are key factors in forming the MRS, but T is a larger impact on the generation of the MRS signal. So let's look deeper inside how the MRS signal changed based on the TE values. The spectra you are seeing now from the same patients and the same anatomical location, but MRS scanned with different TE values. Both TE values are frequently used in the clinical setting. As we can see, these two data show quite different signal patterns. To quickly assess the advantages or drawbacks for the short T MRS, most metabolite signal appears to maximize signal yield. However, the result in the high spectral overlap between the metabolites, making analysis difficult. On the other hand, in case of the long T MRS, certain metabolites can be observed and quantified without the need for the additional analysis tool. However, it inherently has a lower signal yield than short T MRS and may require additional technical optimization of the MRS scan. Therefore, in the neuroscience studies, it may sometimes be more beneficial to acquire MRS data under the short T condition, and it is often necessary to observe the change in multiple metabolites to elucidate metabolic mechanism. In this sense, the following section on the data analysis and AI application will be based on the short T MRS. Now we have covered what elements are included in MRS data, how they are obtained, and how they should be acquired, and what need to be acquired. So the next, how do we analyze the acquired MRS data? Typically, the primary elements for the data analysis in the MRS field can be briefly summarized in the following 4 items. Of this, we have already discussed the first 2 elements, and so I briefly introduce what the last 2 items are about. In commonly, the algorithm utilized for the metabolic quantification is called the nonlinear least square fitting method. And we can describe it in one simple equation that you can see here. That is, for the water suppressed MRS signal, we go through the process of the approximating them by the combining the various parameters, including the metabolite basis set. During this process, we estimate the amplitude for each metabolite concentration. The tool shown below in the research-oriented software for the metabolic quantification developed by the various research groups and have been released after their individual optimization process from this formula. Our basis set, when I mentioned in previous slide, refer to the collection of model metabolite MRS data as shown in the images in here. And basis set can generally prepared in two ways. Either the simulating based on the quantum mechanics, reflecting the molecular structural characteristics of each metabolite, or by using the phantom prepared of the brain metabolite chemicals. It is crucial that the basis set is simulated or data acquired under the conditions that are completely identical to scan parameters of the MRS data to minimize unforeseen error in the analysis. This prepared basis set plays a pivotal role in the nonlinear least square fitting process. The result obtaining using the quantification tool, firstly, unnormalized raw values for each metabolite along with their respective reliability indicators, and in terms of reliability indicator for each quantified metabolites, called Kramer raw bound, are typically used. These CRAB values express the precision of quantification results obtained through the NASF method. However, the utility of CRAB is subject to ongoing debate within the MRS community. Meanwhile, the raw value from the quantification result can be normalized in two ways. First, normalization can be done using the internal reference metabolites such as total creatine amplitude. And this metabolite generally exhibit a quite consistent level in the various physiological conditions, making it frequently used and easily used to internal reference metabolite. However, caution should be needed when applying this approach in case like the brain tumor or energy metabolism related disease. Secondly, water unsuppressed MRS data can be used for absolute quantification. In this step, 3D T1 weighted MRI data like MP-RAGE also can be used to extract gray matter, white matter, or CSF composition from the target voxel. Although this process is more complicated due to the need for additional data types, the advantage is that it allows for expression of concentration directly, making it widely applicable for the various diseases. In summary, for the data acquisition, utilizing short-T MRS is generally advantageous for the complex observation of various metabolites. For the comprehensive analysis, it is advisable to acquire for the both water suppressed and water unsuppressed MRS data. Secondly, for the data analysis, the use of the metabolite basis set is essential when employing the metabolic quantification method for analysis. And the credibility metric like CRAB, commonly used as the reliability indicator, is the subject to debate concerning its meaningful utility. So next, let's discuss the application of deep learning in MRS. This table contains the various AI-aided MRS research topics that have been conducted by our company or during my past training periods at the Seoul National University Hospital. Among this talk will focus on the deep learning-aided metabolic quantification techniques that could potentially improve the conventional metabolic quantification methods. With this regard, I will provide a brief introduction to this DL methods, along with the several evaluation of how they can currently be applied in the neuroscience field. And most of all, I'd like to talk about NMR first, which is the well-established method for metabolic quantification in biochemistry field. The NMR spectrum has excellent data quality, and there is no baseline in there. On the other hand, MRS spectrum, as we already seen, suffers from the low SNR, severe spectral overlap, and most of all, broad-based spectral baseline due to the mobile or immobile protein or lipids. And as a result, metabolic quantification is very challenging compared to the NMR. To overcome the challenge, nonlinear least squares fitting is most commonly used in MRS as talked before, and however, quantification performance is known to be subject to variability depending on the choice of software, pre-processing steps, and quality control parameters. Moreover, the severe spectral overlap of metabolites was occurred inevitably at the clinical MR fields such as 1.5 or 3 Tesla. This issue leads to extremely limit the quantification of key metabolites biomarkers in various brain diseases. Therefore, developing a more robust quantification tool was a remaining issue in proton MRS field. So, recalling to the NMR, if we have a spectrum like this, namely intact metabolite spectrum, we can achieve very accurate metabolic quantification by solving a simple inverse problem. But this is what we have in vivo. So our question is, how do we get this sort of nice intact spectrum from the in vivo spectrum? Since the mid-2010s, there has a steady increase in the research applying the AI to the field of medical imaging. And with this regard, we apply the deep learning to solve our problem. That is, intact metabolite spectrum mining using a deep learning in proton shorty MRS. This slide shows the technical advancement we have made since our initial report in 2019. And to date, a total of 3 technical papers related to quantification have been reported, and we are currently preparing the additional technical papers. And at the current level, the deep neural networks operate around 17 million trainable parameters and has been trained on the more than 10 million in silico brain dataset that closely resemble real-world in vivo brain MRS data. Alongside this, we are also addressing the explanatory limitation of black box AI by presenting scientific uncertainty metrics along with the quantification results. These are the summary of our developed method performances. The metabolic quantification performance in terms of absolute error using the human brain benchmark dataset at 3 Tesla. And as a result, the deep neural network achieved error below 10% for all metabolites, including GABA or glutathione. Therefore, our method allows for research or clinical application based on the neurochemical biomarkers, which was previously only possible with ultra-high-field MR scanners such as 7 Tesla or above. As with any neuroimaging modalities, harmonization of data analysis is essential for the medical application. In this regard, based on our previous studies, we conduct in vivo validation targeting the multi-site and multi-vendor human brain MR spectra. In this process, we analyze in a total of 205 in vivo data, which were acquired from the 9 MRI scanner models and 19 institutions. This graph shows the variability of metabolic quantification in different MRI vendors. Note that our deep neural networks has never seen true in vivo spectra before in a model training phase. However, the DL method nicely quantified the major metabolite compared to the conventional analysis. Given these results, the DL method has more generability in different MR environments compared to the conventional analysis. Next, I'll introduce the age-related metabolic change in healthy adults. The MRS protocol used the conventional parameters on the standard MRI scanner, analyzed the data which were acquired from the PCC region, and in a total of 125 subjects ranging from 20 to 70 years old. These results show the age-related change in participants' NAA compounds, which are the most dominant neuronal activity marker in proton MRS data. On the left, the results with LC model, which is the most widely used in conventional MRS analysis, and right side are the results from our deep learning method. As you can see, the LC model results show a decrease in NA compounds with age, but the scanner harmonization is not achieved. And in contrast, our methods show the decrease more precisely with aging. And moreover, the distinction between the NAA and NAAG, which exhibits severe spectral overlap, is also well resolved through our deep learning method. In case of the glutamine and glutamate, one of the major neurotransmitters, the deep learning method showed a more clear age-related metabolic change compared to the conventional analysis. Next, the total choline, which crucial metabolite biomarker for cell membrane turnover, has interesting results on both methods. This method consists of the two components, phosphocholine and glycerol phosphocholine, and both of which are also severe spectral overlap due to the very similar molecular structure. In this case, the LC model shows that it is impossible to separate these components, but when analyzing total choline together, resulting in a constant value. On the other hand, our method clearly demonstrates the PCH was decreased with age and GPC is increased with age. Although not included here, and an analysis of MRS data obtained from anterior cingulate cortex also shown an equal trend according to the age, and the post-post MRS shown the similar same metabolic trends with this result. Therefore, these two metabolites may potential to cell membrane information about age or neurodegenerative diseases. Similar comparative results were obtained for other metabolites as well. In particular, GABA showed a decrease with age, decreasing trend with age, and myosin is increasing trend. These are consistent with previous results. However, our method displayed more distinct pattern compared to the LC model. The final contents I present involve the functional MRS results related to the pain response. The data for this study was collected at 1 minute interval from the anterior cingulate cortex region while applying the capsaicin and thermal pad to subject to induce temporary pain sensation. The focus of the study is to instant observe change in glutamate level, a representative excitatory neurotransmitter. The MRS scan were conducted using the standard MRS protocol and were performed. In this result, LC model indicates the averaging increase in glutamate level under the pain onset condition in the MRS data. However, there is no statistical significance was observed, and the increase was not consistent across the individual volunteers. On the other hand, our method consistently showed the increased glutamate level for each volunteer and exhibit a strong statistical significance on the average results. In addition, we are currently working with the several other organizations, including the UPM, on the studies related to this disorder. But due to the data security concern, we are unable to share the MR. Nevertheless, deep learning MRS has the potential to be applied to a wider range of neuroscience-related research and clinical application than previously possible. So this is the last slide. In today's talk, we have covered the range of topics from the fundamentals of MRS to the data analysis and the application of deep learning method. To summarize, MRS enable the approximately 20 different metabolites information in specific location within the brain. When it comes to data analysis, conventional analysis method allows for the quantification of only limited set of metabolites. However, the use of deep learning analysis technique can enable more accurate and comprehensive quantification of various metabolites. So with that, I will conclude my presentation, and thank you for listening your attention.
Okay, thank you, Dr. Hyeong, for the insightful and also informative talk. Okay, I will continue from here. So now we open to the audience if they would like to ask for any question. You can type in the chat box, or you can ask directly to Dr. Hyeong. Okay, let me be the first one to ask. I have a question. So when you mentioned about MRS and data analysis using deep learning, so is there any significant difference in the analysis time compared to LC model?
I have to understand what exactly performance compared to LC model. Sorry about that.
I mean, in the analysis time using deep learning analysis compared to the LC model, is there any significant difference in the in the data analysis to be completed? Like any faster?
For the quantification data consistency, or about I didn't data compatibility or the other factors. Sorry, I didn't hear exactly last sentence. Sorry.
When you mention about the deep learning data analysis, right? I mean the time to analyze the data if it's compared to the LC model. Is there any significant difference? Is it like faster?
It is correct. The question is the usage of the quantification tool, right? So actually, as you know, that the LC model is run, first of all, LC model runs on the operating system is based on the Linux. So, but deep learning has the various compatible OS, such as the Windows or Linux or Mac OS as well. So, first of all, accessible to data tool is the more easier to deep learning method. And the usage of regarding the data quantification analysis usage, LC model must be trained for the operating the using meaning, such as as mentioned before in preparation of the basis set, like this. And then there also has the hidden parameters is also in the LC model to more precise metabolic quantification, but it is not exactly explained in the manual. So it is the very high variability, which is the trainable period on the LC model quantification, but deep learning is oriented from the big data from the capturing the feature. So it is used to only the input and output process. So there is no other preparation in the data quantification. You can easy to access you can simply input the MRS data, and the neural net has been output measurement quantification result. Yeah. So
Okay.
there is no additional steps for the data use in deep learning method.
Okay.
Yeah.
Thank you. Okay. Okay, there's a question from Nur Hafiza. Is there a difference in the analysis approach when utilizing MRI scanners with 3 Tesla and 7 Tesla full strength?
Actually, there's no difference between the magnetic field strengths, but as mentioned in earlier, as mentioned in pre, so basis set is must be matched to the MRI scan parameter, like this figure, and a basis set of the 3 Tesla and 7 Tesla is quite different signal patterns with the different due to the different magnetic field strength. So it is separately preparation for the basis set, and other process is quite similar to both field strengths. So yeah. The crucial thing is the basis preparation and match to the MRS MRS scan parameters in each MR scanner.
Okay, there's another question. Is there any difference in the assessment or evaluation using single voxel and multi-voxel MRS?
Well, fundamentally is very similar to the assessing or quantifying the results between the single voxel and multi-voxel MRS, but it is huge difference of the orient of the data acquisition parts, because MRSI, multi-voxel MRS, is the pulse sequence of the multi-voxel MRS is the fusion pulse sequence formed with the MRI and single voxel MRS pulse sequence. So data pre-processing before the metabolic quantification is quite labor intense compared to the single voxel MRS. So it is actually MRSI raw data format is not a 1D signal, but it is from the KT like the KT space. So it is quite more data size compared to the MRI, and reconstructed the MRSI data is quite time consuming to the final quantification result obtained.
Okay, thank you for the answer, Dr. Hyeong. So, next question from Zarina. What kind of chemical phantom has been used in the study? Does it mimic to human brain?
Yeah, it is a very important question. The basis set of chemicals is already selling in the major chemical company like the Sigma Aldehyd or the Merck. So brain metabolites is also same with has already known what exactly molecular structure is formed. So we can buy these chemicals from the chemical company like Merck, and make a phantom with the specific concentration of millimole, like for example 20 or 50 millimole, with the pH level is just blood samples like 7.4, and we can build the chemical phantom like aqueous solution and scanning the MRS scanner with the same parameter scan, same MRS parameter scan with the patient data. So in this step, you can acquire the pure single metabolized chemical data for the preparation of the basis. So it is quite labor intense. Also quite labor intense because it is those chemicals are the individually preparation for to prepare the basis.
Okay, I hope that answers the question. The next question is, what program would you recommend to analyze MRS data?
Okay. So actually, we are recommend to the gold standard conventional analysis, the conventional tool is the LC model, but actually we are also developing the AI-aided MRS quantification tool. So if you wanted to the thorough research collaborator or for the data MRS data analysis, please contact us. Yeah.
All right. Okay. The next question. Do we need to do validation for the phantom mimic human?
It is the subsized validation method, like the in vitro phantom like this. So it is, also building the 17 around the 17 to 20 metabolites mimic chemical phantom is also possible, but it is quite highly cost and then time consuming to make preparation. And crucial thing is the chemical mimic phantom is not reflect the real in vivo data. The crucial thing is the macromolecular baseline. This this baseline composes the mobile or immobile protein. So it is not the modeling or the not specified chemicals in the outside of the brain. So it is really hard to modeling in the in vitro phantoms for the thorough evaluation. So, but yeah, it is quite difference between the in vitro and the in vivo MRS data.
Okay, thank you, Dr. Hyeong. The next question is, what is the minimum possible voxel size for MRS?
Actually, it's very dependent to the what analysis tool will be used. So actually, in MRS consensus, so recommend MRS quantification consensus is recommend the SNR is higher than the 3. Yes, higher than the 3. So it is with regarding this SNR creatine value. Actually, minimum single voxel size actually used at minimum 4 cc, 4 cubic centimeters. Yeah.
Okay, all right. I think that's the last question. Okay, thank you, everyone. So before